Okay, this is our final presentation on our proposed identity and function of our unknown gene. I'm Nicole.
And as always, I'm Andrew. So the first thing that we did was we wanted to get our sequence for our unknown gene. So what we did was we did two different sequencing. We did ab initio sequencing and we did homology-based sequencing. So we got two different results for those two. So we wanted to compare and see which one was correct. So for the ab initio sequencing, we got 7 exons. And from the homology base, we got 9. So in order to compare and see which one was correct, we used Phytozyme's log-scale RNA sequencing data and found that the peaks correlated to our 9 exon proposed exons. So we decided to accept that that was probably the best way to model our gene. So what we then did was we provided the protein sequence, as you see in the bottom right, and annotated that with our different exons. So for exons 1 through 3, they are in reading frame 3, exons 4 in reading frame 2, and exon 5 through 9 are in reading frame 1.
And then we looked further and saw that there are different protein domains that have been conserved. And there's in our gene G-lectin, S-locus, pan domain, transmembrane domain, serine, thurine, kinase domain. And we found the same domains in the OSC2 gene, which is found in rice. And they did experiments with a knockout and overexpressors, and the overexpressing individuals survived at a much higher rate, both under salt stress and drought stress, while the knockout mutants survived at a much lower rate, both under salt stress and drought stress, with only 1 out of 100 and 31 out of 189 compared to a much higher rate with the control in both. And we can conclude that this means that our gene must also be involved in stress response because they share the same conserved domains and must have the same kind of function, hopefully.
All right, so the next thing that we did that we thought was important was to identify the different mutations that can occur within our gene. So we found that there were 9 possible mutations, or there's definitely more than that, but the ones that we found were 9. So we found 7 moderate impact mutations, which are depicted by the yellowish diamonds, and 2 heavy impact mutations, which are pink, which are the red diamonds. So the 7 moderate impact mutations are all missense mutations, which are single nucleotide polymorphisms, changing a single amino acid to another one. For example, the first one, which is an alanine, through to a threonine. But for the heavy impact mutations, so those are also single nucleotide polymorphisms, but they're changing a single amino acid to a stop codon. So that would probably result in a truncated protein. So the information that we got from our professor was the mutation that was actually contained in our mutant plants is the glutamic acid to stop mutation, which is the NAN 1793 mutation contained in our second exon.
And we also looked at our EFP browser data and found that our gene is thermocycled and not photocycled because, as you can see in this graph, when the conditions are hot-cold conditions, there is a cycling nature to the graph, but when it's hot-hot, there's no cycling nature, and this is not affected on the light-dark conditions. It's only the temperature conditions, so we can conclude that it is thermocycled and not photocycled. And to look further on this, we looked at the EFP browser data for an experiment where there was a 7-day cold conditions, which had much higher expression than a 7-day non-cold control. So we can conclude that our gene is expressed at a higher rate under cold conditions, which is a stress, so more stress response.
So after looking at the literature and the EFP browser, we wanted to propose an experiment that confirmed that our gene did in fact have to do something with the abiotic stress response in plants. So we found that salicylic acid produces that abiotic stress response in plants. So we wanted to test this and see if it actually did it in our plants. So what we would do was we would grow a number of our seeds on a petri dish in MS media for 7 days on continuous light at 23 degrees Celsius. And this was done before, so it's a proven method for growing the Brachypodium plants. And then we would transfer them to 2 different petri dishes. One petri dish would have a mock treatment, which is just normal MS, the same thing that it started on. And the next treatment would be a MS media with 10 micromolar salicylic acid treatment. So it's important to do this because so the plants would have the stimulation of a touch. So it's not a lingering variable in the experiment. And then the next thing we would do is have them grow for 3 hours on the same conditions as before. And then we would sample the roots and send them for expression data. And we hypothesized that our expression of our gene would increase in the salicylic acid treatment.
And just a summary of everything, we identified our gene to be Brady1G25180, and we proposed that the resulting thermocycling protein interacts with downstream proteins involved in stress response. And this kind of research is important because knowing about genes involved in stress response can improve production of hardier crops and biofuels, and just general scientific curiosity is really important.
So lastly, we just want to thank our professor, Samuel Hazen, our TA, Katie Webster, and our lab coordinator, Kate Dorfman, for all their hard work this semester. Me and Nicole definitely learned a lot, and we're thankful to all those who have contributed.
Yes, and these are our references. Thank you.
Awesome. Thank you.